Our speaker today is Brandon Rice, who's the Head of Development and Strategy at Dovetail Genomics. And so, Brandon, I'll let you take it from here.
Thank you very much, and good morning, everybody. So I'm going to tell you all a bit about who we are at Dovetail and what we do and how we do it and what kind of results you can expect to see from us. So at Dovetail, sorry, struggling a little bit with the slides here. There we go. We are genome assemblers, and we have, what that means is that we can help out with any or all of the process of getting to a genome assembly, from doing the library prep and the sequencing to the de novo assembly part to the scaffolding. We have services and data for every stage of that. And we can do as much as the entire process, so have folks send us a sample and then return a full chromosome-scale genome assembly. Or if you already have an assembly and you just want to kind of take it to the next level, improve the contiguity or the accuracy, we can help out with that as well. And the way that we do that is we're primarily focused on our proximity ligation data types, which provide very nice information over a range of information scales. I'm going to talk quite a bit more about those here in just a second. And we've been doing this for quite some time. This slide says over 200 genomes assembled. I wouldn't be surprised if that number is closer to 300 now, since this deck was originally made. So we've seen a great deal of the diversity that is out there in genomes, and we've dealt with most of the challenges that there are to be dealt with. And our aim is to make this part of the process extremely easy and streamlined, so that you folks mostly don't have to think about it, and you can focus instead on the biology and asking your questions and getting the resource that you need to enable you to do the genomics analyses that you'd like to do. So to offer a little bit more detail about what specifically we offer in our services, this is our menu. So we can do high molecular weight DNA extraction, which is very important for getting kind of to the intermediate scale of information for doing genome assembly, which is on the order of 100 kilobases or so. We can produce a number of different library preparations, some common and some proprietary. So we can make both Illumina Shotgun and PacBio libraries, which are fantastic for doing the de novo assemblies and necessary for doing the de novo assemblies. And we also produce Chicago and Dovetail Hi-C libraries, which are two different proximity ligation library types, and these are proprietary to us. And I will talk a bit about how these work and the trade-offs of the complementarity between them in a couple slides here. In addition to producing these libraries, we also offer the sequencing, both PacBio sequencing and Illumina sequencing for the Shotgun libraries and for our proprietary libraries. And then we can take those data and do the de novo assemblies with the Illumina data using Miraculous, and with the PacBio data using the Falcon de novo assemblers. And then we scaffold those assemblies, whether we make them or whether you make them, using our proximity ligation data types and our purpose-built proximity ligation scaffolder, which we call HiRes. And finally, at the end of the process, we can use whatever data is available, whether it's Shotgun or full coverage PacBio or low coverage PacBio, to do gap filling and improve the config N50, the contiguity up closer to the level that we can get the scaffolds to. So first we'll talk a little bit about how these proximity ligation methods work and how and why those data are beneficial to the assembly process. So I mentioned a number of times now the Chicago libraries and the Dovetail Hi-C libraries, which the process for which is depicted here in this cartoon, simplified. And the middle part of the process is pretty much exactly the same for both libraries, and where they principally differ is in the beginning with the input material that you're using to start the library prep, and at the end with the aspects of the data that you get out. So first we'll focus on Chicago, which is depicted on the top here. So Chicago differs from Hi-C in that Chicago begins from pure high molecular weight DNA, whereas Hi-C begins from whole tissue or cells. And we'll see why that's important here in a second. So with Chicago, we're beginning with the DNA, which is represented by this black strand over here. And so we'll imagine that this is a very large DNA fragment on the order of 100 kilobases or so. And as we go through this cartoon, we're going to follow the number one and the number 2, which mark the distant ends of this molecule, which in the genome are going to be 100 kilobases apart. And this will help us understand how we can capture long-range information and read it on short-read platforms. So once we have our pure high molecular weight DNA, the first step that we take is we reconstitute chromatin, which entails adding some enzymes and some histones, mixing it all together with the DNA, and the enzymes will reassociate the histones with the DNA. They'll wrap the DNA around those histones. And they'll do that in a sort of uniform fashion, spread out evenly across the fragment. So now in the cartoon we have these blue circles associated with the DNA, and those are meant to represent the histones. These histones act as handles, essentially, for the cross-linking step that's going to come next. So we have now this protein DNA aggregate. The next step that we're going to take is we're going to go ahead and we're going to cross-link that in solution. So we're going to form covalent links among the histones. And then what happens when you do that is something like what is depicted in this part of the cartoon right here, which is that formerly long linear molecule gets condensed down into something that's more globular. And that the very nice effect that has is it brings all of these segments of the molecule that were distant, physically distant previously, into close physical proximity. And so in the cartoon here, the red lines represent the cross-links between the proteins. And so you can see now where previously the number 1 and 2 parts of the fragment were very distant, now they are very close together spatially here. And that is one of the key steps of this procedure. The secondary benefit of doing this cross-linking is that you now have these many links between the proteins that are going to hold this whole thing together through the subsequent molecular biology steps, and that's also important. So now we've got this cross-linked chromatin aggregate, and we're going to go in and we're going to cut that up over here in the next stage with a restriction enzyme that's going to leave some sticky overhanging ends. So where previously we had one long 100 kilobase fragment, now we have several hundred base pair fragments. But their association is retained through the cross-links between the proteins. So even though these are no longer connected through the backbone on the DNA, the associations are still retained. The next step after we've done that restriction digestion is we're going to go ahead and fill these ends in. We're going to make them blunt so that we can ligate them. And while we're doing that, we're going to also add a marker, which in this case is a modified nucleotide that has a biotin attached to it. And that's going to allow us to enrich later for the most informative fragments in the sequencing library. Once we've done that, we go ahead and we perform ligations. And because all of these fragments are held in close proximity through these cross-links, they are much more likely to ligate to one another than just some random fragment that's floating around elsewhere in the solution. And so what you end up seeing are numerous ligations within each of these chromatin aggregates. And the chimeric molecules that these create follow a predictable statistical distribution that we're going to look at here in a second. And that's the information that we're going to use for doing the scaffolding. So you can see here in the cartoon that the number 1 and 2 segments are now joined into this one chimeric molecule. So we've essentially captured information about these formerly very distant parts of the molecule by linking them together into this one sequenceable short chimeric fragment. And once we've done that, most of the work is done now. So we can then go ahead and we can reverse the cross-links and remove the proteins. And now on the far part of the cartoon here, we're down again to just pure DNA molecules. And these are the molecules that are going to go into our sequencing library. And so you can see after we've removed the proteins and cross-links and we've done some end polishing here to remove these biotins that are on the ends of the fragments, the only biotins that then remain are the ones that are interior to the molecules. And so these molecules that contain biotin at this point are those which would have participated in a ligation event back here in the process. And so those are going to be the most informative molecules. And when we do a strip-tabulating enrichment of this library, we're going to greatly enrich for those highly informative molecules here. And then what we do is paired-end sequencing of these molecules. So the little arrows beneath the fragment here represent the reads from either side. So you can see we're getting a read from the number 1 locus, and we're also getting a read from the number 2 locus, which are very close together here in the sequencing molecule, but again, very distant on the original molecule. So we're going to take a look here in just a second at what those data look like, get a sense for what kind of distribution of separations you can see. But before we do that, I'll just point out the differences, or for the Hi-C libraries. And again, all of this intermediate process is exactly the same. So thankfully I don't have to describe that whole thing again. It's exactly the same. The difference is if we start over here with Hi-C with tissue or cells, and instead of extracting DNA, doing chromatin, and then fixing it, we simply fix the native chromatin in situ, in place within the nuclei of the cells. And so we, in a very similar fashion, retain the long-range information by doing that. And in fact, it ends up being much, much longer range than when you extract DNA, because of course, when the DNA is intact, in intact nuclei, it's full chromosomes. So you can get separations up to full chromosomes. So this is how the molecular biology of our library preparations work. And a very important thing, one of the most important things to think about when you're doing a de novo assembly is what's your data recipe going to be. And when you're thinking about that, you want to be sure that you're covering, kind of evenly covering the various information scales that you're going to need to get you there. And generally these fall along short, intermediate, and long range scales. And so to give you all an appreciation of what I'm talking about when I say that, we think of short as basically anything less than 100 kb. So certainly shotgun libraries and mate pairs are short, relatively short, and long reads like PacBio, while certainly much longer than shotgun data, are still relatively short on the scale of genome assembly data types. Getting up into the 100 kb range is things like optical maps, or like 10x, or like Chicago, where now we're talking about information on the scale of hundreds of kilobases, and in best cases, up to several hundred kilobases. And then finally, to close everything out and get up to full chromosomes, you're going to want full chromosome scale information. And currently, the only methodologies really capable of providing that are Hi-C-based methodologies, including ours. So these lines here give you some reference points for where these other data types fall, and what we're going to plot here are some real data from one of our Chicago libraries and one of our Hi-C libraries. So each of these plots is showing the same data. The only difference between the two is the x-axis scale. So on the left plot we're showing up to a 500 kilobase separation, whereas on the right plot we're showing up to a 100 megabase separation. And then on the y-axis is simply the proportion of read pairs within the library that fall at any given separation. So first we'll look at the Chicago, which is the green distribution here. The Chicago read pairs will go out to as far as the DNA, the biggest fragments of DNA that we put into the library prep. So if we put in 100 kilobase fragments, the largest Chicago read pairs we will see will span up to 100 kilobases in the genome. And it follows this, they always follow this sort of characteristic decaying read pair separation distribution. And so what that means is within these libraries you have the vast majority of pairs are at relatively short ranges on the order of single kilobases. And then fewer of the read pairs are at medium ranges at tens of kilobases, and then fewer read pairs still are at hundreds of kilobases. And it's the shape, taking advantage of our knowledge of the shape of this distribution that allows us to scaffold with these data types. And very nicely, essentially all insert sizes between 0 and the size of the DNA that you put in will end up being covered by these libraries. They're quite flexible and powerful for doing scaffolding. So that was the Chicago library here in green. Also plotted is in purple is one of our Hi-C libraries. And you can see that whereas the Chicago, in this case, drops out, looks like, kind of around 250 kb, probably a little sooner than that. And when I say drop out, I mean it flattens out here, and at that point we're just getting into the noise, and we're not seeing informative fragments anymore. So in the case of this Chicago library, it's likely that the DNA that we put in was 150 or 200 kb in extent. Now if we switch over to looking at Hi-C, we can see that this is extending well out past the end of this plot here, which is the reason why we have this other plot on the right. And you can see that this continues to fall and doesn't flatten out all the way out to the maximum extent of this plot, which is 100 megabases, which is the size of full chromosomes in this organism, which is a human. So that's what's fantastic about the Hi-C, is that you get up to the full chromosome length information. And you can see that it's quite a bit bigger than all of the other scales of information. This black line here indicates the maximum extent of the left plot there. So those are the information types that we have. As I said, it's important to at least cover some kind of short range, some intermediate range, and then some long range to get the highest quality results. And so I'm going to show now an example of hitting each of those marks with a specific project. This is a project that we did with Brant Faircloth at LSU. This is a bobwhite bird, and this is a soup to nuts project for us, where Brant sent us the tissue and we did everything involved, from the shotgun libraries to the sequencing to the de novo assembly, and then the Chicago and Hi-C libraries and scaffolding with those. And so what I'm plotting here are, in each of these cases, a number of scaffolds. So on each of these plots, the little boxes that you can see represent a scaffold. And then the color within the boxes represents some density of Hi-C read pairs. And the reason that we add those in there is just to kind of give indications of how things are going to be coming together. The broad message here is we begin over here with the de novo. This is made from a shotgun library. And you can see that each of these boxes is very small. They're a bunch of tiny little scaffolds. And you can see the scale here only goes out to 80 megabases, just because we have to zoom that far in to be able to see the individual scaffolds. And as we progress through Chicago and then the Hi-C, you can see that the scaffolds get bigger and bigger. So over here, they're very small. Once we go in and add the Chicago, you can see now that things are beginning to come together. And now we're on a 1.4 gigabase scale. That's the whole genome size in this case. And you can see we've got some nice big scaffolds after we add Chicago. So a really fantastic improvement over the de novo. However, so you can see, because this is the Hi-C data being plotted, which at this stage hasn't yet been used for scaffolding, you can see the links that the Hi-C information is going to be telling us to make. So for example, there's this very high density of read pairs in this box right here that suggests that this scaffold is linked to this scaffold. Chicago doesn't know that, but Hi-C does. And once we get over here to the final scaffolded assembly, at this point we're now into full chromosomes. And so whereas in the Chicago assembly you can see there's a lot of sort of off-diagonal, very high densities of read pairs, that's signal that can be used for scaffolding. Over here we've taken advantage of that, and we've strung everything together. And so you can see that in this case the biggest scaffold here is a full chromosome, is 200 megabases. And in fact, each of these scaffolds is a full chromosome. And while there are many of them, we do know that they are all full chromosomes. Birds are known to have a number of these very, very small chromosomes, microchromosomes, which is what each of these small scaffolds are here. So in this case, we went from tiny little shotgun contigs and scaffolds over here, improved those to a nice starting place for the Hi-C library using the Chicago data here, and then got those up to full chromosomes by adding the Hi-C data over here. And this was all done in, I believe, 8 weeks. So not only very nice, high-quality assembly, but also very quick. So that's an example of the whole process for us with a bird. As I mentioned at the beginning, we've done this for many organisms, pretty much all of the broad categories that you could imagine. We've got a reptile over here where These are all the same types of plots I was just showing you, where each box is a scaffold, and in these cases, these all represent full chromosomes for these organisms. And then along the tops, I'm showing the progression in N50 from the starting point to adding Chicago to adding high C. So for this reptile, you can see we began with a very nice assembly at 500 kilobase N50, added Chicago, got that up to 15 megabases. And then when we add high C, we get that up to 90 megabases, which is a meaningless statistic at that point because everything is in full chromosomes. And so here we've got 22 chromosomes, 22 scaffolds for the 22 chromosomes of this reptile. Here a fish, I apologize for the quality of the plot here. 24 kilobases, so a much more fragmented input assembly than either the reptile or the plant on the right. Nonetheless, we can still work with that. We scaffold that with the Chicago library, again, to get it to an intermediate scale of 12 megabases. And then when we add the high C library, that takes us up to 41 megabases. And the final example here is a plant, it's a lettuce. Again, similar story, 500 kilobases, lots of blood, sweat, and tears just to get to this point and many years. We add to that the Chicago library, which is 6.5, gets us to 6.5 megs. And then with the high C, we're at 295 megabases. Again, full chromosomes, 9 scaffolds for 9 chromosomes here with the lettuce. So the last bit I will touch on here is just to talk about the complementarity of these two data types. I've been hammering a bit throughout this on how important it is to have short, intermediate, and long-range information, and this slide here is meant to give a visual appreciation for why that's the case. So what we're showing here is real Chicago data and real high C data being mapped to a scaffold. So each of the purple dots in these plots represents a single read pair. And the position of the dot along the x-axis is the midpoint of where that read pair maps to the scaffold. And the y position of the dot indicates how far apart the reads within the pair map. So if the two reads within a pair map close together, the dot will be close to the bottom. If they map far apart, it'll be further up towards the top. And you can see, as you would expect from those separation distributions that I showed you earlier, there's a relatively high density of information at short ranges or close to the bottom in these plots, and then that fades as you get further and further away. That means fewer and fewer long-range read pairs relative to the other types. So that's how these plots work. We're showing the same scaffold, the same data, the Chicago data on the left two columns, or the left column, and the high C data on the right column. Along the top, we're showing those data mapped against the scaffold where all of the contigs within the scaffold are in the correct orientation. On the bottom, we've taken one of the contigs within the scaffold and we've inverted it. So this is a 50 kilobase inversion of one of the contigs within the scaffold. And the aim here is to see how easily we can detect that inversion with the two data types. So everything's correct up top and there's an error down bottom. So I hope it's fairly evident if you look hard where just exactly that error is in the Chicago data. It's fairly easy to see by the displaced read pairs, which is right there. So you can see if you compare the top to the bottom, there's this unexpectedly high density of read pairs that are kind of raised high up above the x-axis here. And that means you're seeing a lot of read pairs that are further apart than you would expect them to be, which is a classic signature of an inversion within your scaffold. So this is the type of signature that shows us that there's a problem here. These two red lines indicate the breakpoints of that 50 kilobase inversion to show you folks where things are. The exact same inversion is also present over here in the scaffold of the high C data. But as you can probably very easily tell, it's incredibly difficult or impossible to spot the signature of that same inversion using your eye. And in fact, our eyes are quite good at finding these kinds of patterns, and if our eyes can't see it, it's also going to be difficult for the algorithms to see it. So the point here is, when you're going particularly from more fragmented input assemblies like those that you would generate with shotgun data alone, it's very important to add an intermediate scale of information for your scaffolding so that you don't make these types of mistakes. If you jump straight into high-C scaffolding with the shotgun library, you're going to have, it's very likely, and we've observed, that you're going to have lots and lots of small local scale errors in the order and orientation of your scaffolds. And so if you proceed with adding the intermediate scale in, you can largely head off a lot of those problems. So thank you all for your attention. I'm going to go ahead and now turn it over to Brenda to talk about her projects.
Okay, and Nikki, you're going to advance the slides for me, I'm understanding, so you can go ahead and go to the next slide. I think most of you know me and know what I've been working on, but I wanted to just give you just a brief reminder of what I do and why I do it, and then go into some of the data that I've obtained from Dovetail assemblies. We are tasked in our CRIS with minimizing the impact of stored product insects because it's a huge economic problem, not in this country, but more so in other countries where they have less structure to control these insects. Next slide. One of the areas that I've been focused on recently has been looking at insect resistance to insecticides, and primarily to phosphine. Phosphine is the most economical and effective way that we can control stored product insects in storage, and the problem is, like with everything else that's under strong selection pressure, you rapidly evolve resistant insects and resistant populations, and we're seeing that worldwide. So this is a big problem, because if we don't find a way to control this or change our IPM programs, we are going to lose phosphine as an effective control method, and we're going to have to go to more expensive and probably less effective ways to control these insects. And so our program is looking at what is the molecular basis for this resistance in the insects that we're seeing. So that's why we need genomics, and I think that's on the next slide, Nikki. Because the genomics will help us get at some of the questions that we're asking, and help us in some of the RNA-seq studies that we're doing. And in addition, it could probably help us to connect more of the biology of the insects with the, with the genetics that underlie that, and perhaps even find new targets for control. Next slide. So I think the next slide is where we actually, where I began working in functional genomics, and that was with the Tribolium Genome Sequencing Project. I got in on the tail end of that project in annotating genes, and quickly realized that this was the future, so switched from the more protein work that I've been doing into functional genomics. Next slide.
Could you please speak into the microphone? We're having trouble hearing you.
So I'm speaking into my phone because I dialed in, Glenn.
Oh, yeah. Well, you were muffled there when you were talking sideways or something.
Oh, so is this better?
Perfect.
Okay, I'll try to do better. So this is lesser grain borer, and this was the first project that I took leadership of. It is an internal feeder, and so it is more difficult to control. It's more difficult to study because the immature stages feed within the grain kernels. And so we started this project, I think maybe 2007. So 10 years ago maybe, looking at how can we get the perfect assembly. And so we inbred for more than 20 generations, trying to get the most homogeneous population we could start with. And we started sequencing. We had a PGM at the time. We now have a MiSeq. We've outsourced HiSeq and PacBio. Tim Smith at the Lab and Clay Center helps us with that, and we have a collaboration with him. And so we started trying to do assemblies and found that the SoapDeNovo worked best with our MiSeq data, and quickly tried different algorithms for the PacBio data, and we have migrated to Canoe because it gives us fairly good assemblies with a lot less computer power. But then, you know, we reached a certain point, and we wanted to see if we could get even better with the Dovetail assemblies. So that was our first assembly with Dovetail. Next slide. And so this is the kind of representative data that you get from Dovetail when they start talking about the results that they've gotten. And so the Chicago coverage, which Brandon explained, the Chicago libraries, was about 134x with sequencing HiSeq. And we were able to go from a N50 of about 100 that had about 158 scaffolds in it, and about 871,000 bases, and then reduce that down to 20 scaffolds with a 7 megabase N50. And so we were pleased with that because this was our initial assembly, our initial results, and we got much longer pieces of the in the assembly. And so I'll just walk through the data here. So next slide. These are the kind of contiguity plots that we get that show the difference between our assembly, so the input scaffolds in red, and the scaffolds that we were able to get with Chicago Library in blue, and showing that we're getting much longer contigs or scaffolded contigs. Next slide. And this just is a insert distribution plot showing how the distance in base pairs between the insert sizes that we're getting in the Chicago libraries. Next slide. And so we took this data. So we took the assembly that Dovetail gave us, and we went back and grabbed our MiSeq data again because we have more confidence in the accuracy of that data. So we did a hybrid assembly. And so we went from where we originally had the Canoe data that was PacBio only, and we had about 1,800 contigs. And then Dovetail was able to improve that to about 950 contigs. And then our hybrid assembly was able to reduce that by about a third. And we got increases on the average length of the contigs at each step. And the way that we did this was using Sequin Engine in a hybrid assembly of the Dovetail contigs with the MiSeq data. And we looked at the BUSCO metrics for using just Insecta for the reference genes at each step, and we have relatively complete as far as gene the genes that are there. It's relatively a complete assembly, and we took Augustus and found about 23,000 genes in a massed assembly, and so that was reasonable from the standpoint of when we had looked previously, we had way too many genes, and so we had problems. And the next slide explains what those problems were. We can go to the next slide. And that is the repeats. And I've been working with a number of people. This is data from Anna, and I'm not going to try to say her last name, but she is at the Polish Academy of Sciences, works in plants. but she agreed to take a look at our data and just tell me what she saw. And it was quite interesting because we knew already that we had a large number of repetitive sequences, about 40% of the genome. And about 36% of those were transposon related, but only 9% were similar to typical transposons. And so she thinks that these are remnants of ancient transposons. And when she did an analysis of the different kinds of TEs, she found that mostly they were the line and LTR retrotransposon families. And interestingly, there were about 119,000 remnant copies and about 7,000 actual copies of the TC1 Mariner family. But what this data told us was that we had a really diverse TE landscape, and so we think that that was, some of the major problems we were having in assemblies in the beginning, and we think that Dovetail technology can deal with that and give you still longer contigs, longer scaffolds. It doesn't, these repeats don't affect that. Next slide. So I think that this is the next insect that we've worked on with them, and this insect, we started out thinking naively that this is a proof of concept that we could just take a single pupa and extract genomic DNA and avoid those years of inbreeding, and we could get a good assembly from that. So we extracted from a single male pupa, and then we sequenced on MySeq and also PacBio. We had 45x coverage, and we tried to use Canoe to assemble that data. And after days and days, and it was going into weeks of compute time, and we were getting terabytes of data written to the hard drive, we contacted Sergey Korin, who is one of the developers for Canoe. And he was able to get an assembly, but he had to modify his program to suppress repeats, and it still took about 20,000 CPU hours, similar to what is necessary for a human genome. But this is not that large, so we were puzzled by what the problem was. And in the next slide, I was able to determine what the problem was by taking one of the PacBio, or some of the PacBio reads, and blasting to NCBI. The little pink dots all along this are these satellite repeats that are highly homogeneous, and they are found dispersed seemingly randomly throughout most of the reads that I was blasting. And then in the next slide is the paper that I found that explains this, and that was a study that found that these highly similar repeats of 142 nucleotides, less than 2% divergence in 18 different varieties, comprises about 60% of the genome, and it's present in every chromosome. And so it's an interesting question from evolutionary standpoint, but it was a nightmare for these assembly algorithms. And so we went to Dovetail with this information, and I asked Brandon if this was going to be a problem, and he said, I don't think so. So in the next slide, you'll see the results that we got from that assembly. So we had, for our Chicago coverage, we had 53x coverage. We had an L50 of 908 scaffolds and an N50 of about 104,000 bases, and we were able to reduce that to 58 scaffolds with an N50 of about 2 megabases. And so we were pleased that they were able to get this kind of improvement in the assembly, even with these repeat sequences. And that's because, as you saw from Brendan's explanation, the long read data information is important, especially in this case. So next slide. This is the contiguity plot of of that data showing the improvement from our assembly, which was the blue, to the Dovetail high-rise assembly in the red, showing increased scaffold sizes. Next slide. And again, the library size distribution that was in the Chicago library. Next slide. I'm just showing you some of the representative data that you get in your report after you've completed a project. And now they also give you the BUSCO information, so that is included in the report. So next slide. We took that then and did the same thing with it that we did with the Lesser grain borer, and we then took our canoe assembly, where we had about 7,500 contigs. The Dovetail improved it to about 2,400 contigs that were much longer. And then our hybrid was able to about half that into about 1,300 contigs, increasing the length of each scaffold. And the BUSCO metrics, again against insecta only, indicated that that we had a fairly good assembly with most of the reference genes represented. And we had started with Augustus. We don't have a maker program yet that's functional at series, and so we've been using Augustus, which is in the Blastigo package. We are hoping that we're going to be able to get that maker program working so that we can actually use that in gene prediction for these. So that's why we're using Augustus. And we were working on masking and also doing gene prediction, but I went to the users group meeting for Dovetail in June and saw all this beautiful data from the High-C complementing the Chicago. So we have contracted with Dovetail to do this insect and the other insects as well, but we're working right now. We've already sent them pupae for them to extract and make the High-C libraries, and so they're working on this right now for the High-C library, and we're hoping we can get down to close to chromosome scaffolds. So I think the next slide is my last slide, maybe. Or maybe it's the summary slide. Oh, no, this is the next one that we're working with for the Chicago assemblies. But as I said, we're going to go ahead and go to the High-C assemblies, combining with that. Tim is right now working on getting the PacBio sequence that we need in order to give Dovetail the draft assembly going in. This is also an internal feeder. It's a lot harder to work with as far as extracting genomic DNA, and also the genome size is estimated to be a lot larger. We're expecting we'll have problems with repeats. We don't have any information on that going in, but I wouldn't expect it to be any different. So next slide, I think, is the summary slide. So we've been really pleased with the data that we've gotten just using the Chicago libraries thus far. We've been able to reduce the number of contigs to up to 3.2-fold and increase the N50 up to 19-fold, which is helping us greatly in order to do accurate gene prediction. And we are pretty happy with being able to combine Sequment Engine with the MySeq data in order to even get better metrics, we believe. And the BUSCO scores were good, and we think that the Dovetail technology is particularly suited to work with these complex repetitive genomes that many of you are working with, I think, too. And so the last slide, I think, is just an acknowledgement slide of everybody that when you work on a project this large, the repetitive sequences, some of the questions that we're asking, which are not so much concerned with insect control and trying to develop good insect control products, but some of the really interesting questions that these repeats are posing, and we're hoping we can get some help on this to really do a good job on trying to explain how these things have expanded and why they're retained in the genome. So the last slide has to be the acknowledgement slides. And these are the people that have worked with, on the project. Chris, Ken, and Tom, and have been working on the inbreeding and also the extraction of the DNA. And Tim, as I've already said, has worked with us on the PacBio sequencing, and Sergey has helped us with the assemblies, as has Richard Hall at PacBio on the assemblies. And then Anna, I've already spoken about, but in particular, working with Ed, Brandon, and Margot was our point contact. So when you work with Dovetail, they will give you a person who is your person who you communicate with and who watches your project through all the phases. And that's really helpful because you have a person that you can immediately contact and get information about your project. And Mark is great at getting you set up and giving you quotes and figuring out what you need. So it's been a great team to work with, and I've enjoyed it. So thank you. I guess we'll answer questions if we have any. I'll go mute right now and just wait unless I'm asked a question.
Yep. Sorry, we're having trouble unmuting here. Wonderful. We've got several questions already. I'll go ahead and...
Well, it's Kevin Hackett. Hi, Brenda. I was just wondering, the incredible proportion of satellites, does this have any has anybody tried to see if there's a phylogenetic relationship to abundance of satellites? And is there any, even within the beetles, is there any relationship to other clades?
Unmute yourself.
You talking, Brenda? Because we don't hear you. Somehow I think.
All right, we'll work on that mic issue. I'll go ahead and ask Brandon a question here, and Brenda, let us know if we can hear you while you're working on that. There was a question about how much DNA or tissue is necessary in order to make Chicago or Dovetail Hi-C libraries.
Yeah, so for the Chicago libraries, we look for 500 nanograms of DNA, high molecular weight per library. We generally like to have a couple multiples of that so we can try things out if a first attempt doesn't work. We also prefer to do the DNA extractions ourselves because we've become quite good at getting this high molecular weight DNA out of the samples. But that being said, 500 nanograms per library. For the Hi-C libraries, it's quite a bit more variable because it depends on the quality of the tissue and the nature of the tissue and so forth. But they're generally just, a couple of grams is usually sufficient. It depends, though, on how woody the tissue is or cell dense. But be happy to provide much more specific sample guidelines if someone wants to reach out to our info line.
Great. Can you give us a general ballpark about the cost for library preparation and sequencing?
Yeah, sure. So the way we quote our library preparation services is we don't really charge by the library because these are complex projects and we do what it takes to get the number of libraries that we need to scaffold the genome. So the way that we quote is by genome size instead. And we do have a formula that corresponds to a certain number of libraries that scales with the size of the genome, but that's not important to the customer necessarily because we're going to make as many libraries as we need to. It also depends on how much of the project folks want us to do, whether they want us to do the de novo part as well as the scaffolding or just the scaffolding. But so just to give some ballparks around those, if we're talking about, say, a 1 gigabase genome with library generation and sequencing and all of the informatics and QC and so forth, we're probably looking at in the neighborhood of 10K for a scaffolding project, and that goes up to probably about 20K for a de novo project. Those are very, very, very rough estimates, and don't hold me to any of that, but just to give folks a rough idea of where the costs for those land.
Great. Can you talk a little bit about how well the Chicago and the Hi-C approaches handled a heterozygosity and structural variance, especially when you're talking about needing, possibly a gram of tissue for a Hi-C library that's probably going to involve sequencing a population of insects rather than a single insect?
Right, that's a great question. So in the worst cases, so the place where variation is most challenging is at the de novo stage. Typically, if we can get a decent de novo assembly, and when I say decent, I mean assembling over 75% of the non-repetitive content and getting that into pieces that are with an N50 of 20 kb or bigger, that's a nice starting place for us. If we can get to that point, for the most part, these structural variants don't have a major impact on the scaffolding capability. What ends up happening in most cases, in the best cases, is that one of the two variants gets picked and represented in the final assembly. So imagine the case where you have like a megabase inversion, one of the chromosomes, and half of the genomes in your sample have it and half don't. In the best case, one of those two alleles gets picked and represented because, of course, all of these assemblies are haploid. In the worst case, there's kind of a dead even tie in terms of the amount of data supporting the two possible choices, and then no choice is made and no join is made in those cases. So even in the worst cases, we're not going to be introducing errors into the assembly. We'll just not be able to make a join, but that is a minority of the cases.
Okay, and are the sequencing kits available for library prep, or do we have to send DNA or tissue to Dovetail? I know you said you may have some preference, but do you have those kits available?
Currently, and particularly the library preparation, is only offered as a service. And in addition to that, we can also do the DNA extractions, but the customers can do that. Customers can do sequencing. So the only part that strictly has to be performed at Dovetail by Dovetail are the Chicago and Hi-C library preps and the high-rise scaffolding.
Brenda, have we been able to get you back online? Are you able to get unmuted?
Yeah. Can you?
Okay,
so now
great.
I'm on my. So now I'm on my web phone.
Okay. Great.
Can you hear me?
Were you able to respond to Kevin's question? I don't know. Do you need him to re-ask?
No, I couldn't hear Kevin's question. So can you ask it again, Kevin?
I was just wondering about the incredible surfeit of satellite sequence, repetitive sequence in the clade you're working with. Is that typical of phylogenetically and within the beetles or in other groups? I mean, is anybody trying to put an evolutionary explanation to it?
Yeah, there are some groups looking at this, and of course tribolium is in that, or tenebrionids as well. You don't see it to that extent in tribolium, and they're different. So the repeat sequences, but I didn't realize this, but tribolium is not a complete genome sequence. These repetitive sequences have never really been sequenced through. So what we're working with as the reference genome is just the stuff that we could sequence.
So you might need Dovetail for that, huh?
So
I think it would help.
This is Keith Hopper, and this amount of repetitive DNA, particularly in relic transposons, is really common. Like in Lepidoptera, the Lepidoptera that have been studied, like Bombyx mori, like 60% of the genome is repetitive DNA, and a lot of it is relic transposons. It's also true in plants. And these relics have effects on fitness and are often involved in rapid evolution.
Yeah, Keith, we've been interested in it from a standpoint of gene duplication, and we think it could be helping to rapidly duplicate some of these genes that are involved in detoxification or, specialization of some of these enzymes for certain diets, things like that.
Well, not only that, I mean, at least for BT resistance, there's been a loss of target site through insertion of a transposon in the cadherin gene. So, I mean, at least in those, that's the case. I mean, it seems to me insecticide resistance, especially major gene effect on insecticide resistance, may be mediated by transposons.
Right. I agree.
So we have another question here. Brenda, can you explain if you expect the total genome size to be reduced in both of your example species when the hybrid assembly is performed after dovetail?
I do think that it will be, because we're coming in about 490 megabases right now, and the genome size was predicted by I think a number of different methods. We're actually working right now to have it accurately predicted by Spencer at Texas A&M with a laser, but it's estimated to be about 479 megabases, so we'll probably reduce that a little bit.
Okay, wonderful. I think with that, I realize that we're reaching the end of the hour here, so I want to thank everyone for calling in and thank our speakers, Brandon and Brenda. And I just want to say, if you're interested in enhancing your research with Dovetail's genome assemblies, I'd really encourage you to visit their website, dovetailgenomics.com. And currently they've got a 2017 Research Impact Award that you should really look into. The winner will receive a free genome assembly, and 5 runners-up will receive discounted services. So the submission deadline for that is really coming up soon. It's August 8th, and the winner will be announced on August 10th. So please take a look at that. And with that, I just want to say, tell everyone, please have a great day, and I hope you'll join us next month for a presentation by Illumina's Applied Genomics Group. Thank you so much.
Thank you all very much.